Open a heavy archival drawer in the Bayerische für Paläontologie und Geologie, the Munich Museum. Inside, beneath layers of protective tissue paper, sit black and white photographs printed over a century ago. The edges are curling, the sepia tones fading into a muddy brown. The images show a set of massive bones laid out against a simple scale bar. They are vertebrae, the interlocking blocks of a backbone. But extending upward from each one is a towering, flat blade of bone, some reaching nearly 2 meters in height. This is not a reconstruction or a drawing. This is the photographic record of a physical object pulled from the Sahara Desert in 1912. To understand the animal that left these bones, we have to look past the fading paper and orient ourselves in deep time. We are looking at the late Cretaceous, about 95 million years ago. North Africa was not a desert then. It was a vast, humid, shifting river delta environment bordering the ancient Tethys Sea. The air was thick, the waterways broad and choked with life. And moving through this landscape was a predator unlike any other. The fossil collector Richard Markgraf extracted these bones from the sandstone of Egypt, wrapping them carefully for transport back to Germany. In 1915, the paleontologist Ernst Stromer published a detailed monograph describing them. He looked at the elongated lower jaw, the unusual conical teeth, and those extraordinary, towering neural spines forming a massive sail along the back. He named the animal Spinosaurus aegyptiacus. SRC01, Stromer. 1915, for decades. Those bones sat mounted on metal armatures in Munich. They were the physical baseline. In science, when a new species is named, the original specimen becomes the holotype. A holotype is the gold standard against which all future discoveries must be measured. If you find a bone in the desert and think it might belong to Spinosaurus, you travel to Munich, pull out your calipers, and compare the ridges and muscle scars of your new find to the holotype. That is how the system of paleontology works. But on the night of April 24, 1944, a light bomb struck Munich. The museum was heavily damaged, and the holotype of Spinosaurus aegyptiacus was reduced to ash and rubble. The physical baseline was wiped out completely. The destruction of a holotype is a permanent scar on the scientific record. For the rest of the 20th century, Spinosaurus became a ghost. We knew it had existed because Stromer's incredibly detailed sketches and the sepia photographs survived the war. But drawings cannot be placed into a CT scanner. Photographs cannot be sampled for chemical isotopes. As paleontology advanced, developing new tools to peer inside bone and extract the microscopic secrets of extinct life, this animal remained locked in 1915. It was a silhouette, an outline waiting to be filled. To fill it, field crews had to return to the Sahara. The rocks that yielded Stromer's original bones extend far beyond Egypt. They stretch thousands of kilometers west across North Africa, terminating in the dramatic, sheer cliffs of eastern Morocco. This is the Kemkem group. It is a harsh, unforgiving landscape of layered red and yellow sandstone baking under the modern sun. But hidden within these vertical rock faces is the compressed sediment of that ancient river delta environment. Fieldwork here is not a matter of dusting off perfectly articulated skeletons laid out on flat ground. It is an exercise in vertical extraction. Researchers and local miners hike up steep scree slopes, clinging to the cliff face, chipping away at the crumbling sandstone with rock hammers and awls. When a fragile bone is found, it must be stabilized with liquid consolidant, wrapped in a protective plaster jacket, and lowered carefully down the escarpment. It is exhausting, dangerous work. And what comes out of the rock is profoundly frustrating. The Kemkem group yields fossils, but it almost never yields whole animals. What we find are fragments, a piece of a jaw, a shattered rib, and above all else, teeth. Thousands upon thousands of teeth. The sandstone is packed with them. If you sift through the eroded sediment at the base of the cliffs, you will inevitably find long, smooth, conical teeth. They lack the serrated, steak knife edges of typical meat-eating dinosaurs. Instead, they are engineered for piercing and gripping. They are the unmistakable signature of Spinosaurid dinosaurs. Much like modern crocodilians, shed and replaced their teeth continuously throughout their lives. A single animal could leave thousands of teeth in the river mud over its lifespan, while leaving only one skeleton when it finally died. The sheer volume of these teeth in the Moroccan rock is staggering. These teeth are direct evidence that Spinosaurus aegyptiacus was here, hunting in the ancient waterways of Morocco. But a tooth alone cannot tell you how an animal stood, how it walked, or how it swam. An abundance of teeth simply proves existence. It does not solve the architecture of a body plan. And as researchers continued to pull these isolated fragments from the Moroccan rock, they realized they were facing a problem much larger than just rebuilding a destroyed dinosaur. The cliffs of the Kemkem were hiding an ecosystem that, according to the standard rules of biology, should not have been able to exist at all. Stand at the base of the Kemkem escarpment and trace your hand across the exposed rock. If you look closely at the cliff face, you will notice that the bands of red and yellow sandstone do not lie perfectly flat, stacked like a neat layer cake. Instead, they swoop and intersect. Thin lines of sediment cut across one another at harsh angles, creating a woven, braided pattern in the stone. Geologists call this crossbedding. It is the unmistakable fingerprint of moving water. This simple visual cue tells us that the late Cretaceous Sahara was not a static lake or a quiet floodplain. It was a high-energy environment. Channels of water carved through the landscape, changing course, flooding their banks, and scouring out new paths. This geological reality presents a massive problem for paleontology. High-energy rivers do not bury animals gently. When a creature dies in a quiet lagoon, it might sink to the anoxic bottom and be covered in soft mud, preserving its skeleton in perfect, articulated order. But when a massive dinosaur dies in a rushing river system, its carcass is battered. As it decays, the bones are pulled apart. The current rolls the skull downstream, snaps the ribs, and scatters the vertebrae across kilometers of riverbed. To find a fully articulated skeleton in this kind of sandstone is statistically improbable. What the field crews extract, instead, are thousands of isolated pieces. When researchers lay these scattered pieces out on lab tables, organizing them by shape and species, a deeply unsettling pattern emerges. Pull out a museum drawer containing Kemkem fossils, and you will find it overflowing with the long, conical teeth of Spinosaurus. In the next drawer, you will find hundreds of the flattened, serrated, blade-like teeth of Carcharodontosaurus, a massive terrestrial predator that prowled the same region. You will find skull fragments of giant crocodilians and the barbed rostral spikes of prehistoric sawfish. What you will struggle to find are the herbivores. In any functioning ecosystem, the pyramid of life requires a massive foundation of plant eaters to support a tiny apex of meat eaters. Walk across the African savanna today, and for every lion, you will see hundreds of wildebeest, zebras, and antelope. The food web demands it. But the Kemkem sandstone shows the exact opposite. The rocks are choked with the remains of giant carnivores, while plant-eating dinosaurs are practically invisible. When Ernst Stromer first recognized this imbalance in the early 20th century, it defied all biological logic. It became known as Stromer's riddle. How could an ecosystem support a population consisting almost entirely of apex predators? To solve Stromer's riddle, we cannot just dig up more teeth. We have to understand how the Earth acts as a sorting machine. Paleontologists rely on a subdiscipline called taphonomy, the study of everything that happens to an organism between the moment it dies and the moment it is fossilized. If we want to know why the Kemkem rocks look the way they do, we have to model the ancient river, in laboratory flume tanks. Researchers run water over different shapes and densities of bone and teeth to see how they behave in a current. The results reframe everything we see in the Moroccan cliffs. Heavy, dense objects, like the thick conical teeth of Spinosaurus or the solid crowns of crocodilians, drop out of the water column quickly. They sink to the bottom of the deepest river channels and become trapped in the gravel. Lighter, hollow bones, or the spongy vertebrae of terrestrial herbivores, catch the current. They are swept downstream, battered into unrecognizable dust, or washed completely out to sea. The river acts as a physical sieve. The overabundance of predators in the Chemchem group is an illusion. It is not a biological reality of the late Cretaceous, it is a geological sorting mechanism. But the river distorts more than just the ratio of animals. It also distorts time. When we point to a specific horizontal band in the Chemchem stratigraphy and extract two teeth sitting just centimeters apart, it is deeply tempting to imagine those two animals crossing paths. We picture a Spinosaurus and a Carcharodontosaurus locked in a territorial battle on the riverbank. But the evidence cannot support that cinematic assumption. River systems are prone to a phenomenon called time averaging. A river might lay down a bed of sand over a century, burying a handful of bones. Then, a massive flood might scour that sand away, churn up the buried bones, mix them with the remains of animals that died that very morning, and bury them all together in a new deposit. This cycle of burying, eroding, and reburying compresses vast stretches of deep time into paper-thin layers of rock. Two teeth sitting side by side in the Chemchem sandstone might belong to animals that lived tens of thousands of years apart. They never saw each other. Understanding taphonomy and time averaging places a severe constraint on what we can claim about this ancient world. The ecosystem we are trying to rebuild was fragmented by water and compressed by time. This means that if we want to reconstruct the body plan of Spinosaurus aegyptiacus, we cannot simply walk along the cliff face and hope to find a pristine skeleton waiting in the rock. The geology forbids it. This geological frustration is compounded by a modern human reality. Because these fossils are scattered and buried in steep, dangerous terrain, traditional scientific expeditions rarely find the best material. Instead, the local economy has adapted. For decades, local Moroccan miners have dug deep, narrow tunnels into the Chemchem escarpment, extracting isolated bones and teeth to sell in the commercial fossil trade. This is the Moroccan fossil bazaar. When a scientist buys a cardboard box full of dinosaur bones from a commercial dealer, a critical piece of scientific data has been erased. The exact provenance is gone. We do not know exactly which layer of sandstone a bone came from. We do not know what other fossils were sitting next to it. We just have a bone, divorced from its context, floating in a vacuum. If river currents scatter the bodies, and commercial mining erases the exact location of the fragments, piecing together an extinct animal becomes a daunting puzzle. We must find a way to trust unprovenanced bones. We must figure out how to prove that a jaw fragment and a leg bone, pulled from the vast, chaotic expanse of the Sahara, belong not just to the same species, but to the exact same individual. The assembly of a dinosaur from scattered, unprovenanced fragments requires a method capable of looking past the surface of the bone. In 2014, a research team led by Nizar Ibrahim presented a new partial skeleton of Spinosaurus aegyptiacus, pieced together from material acquired primarily through local Moroccan fossil hunters. This specimen, catalogued as FSAC KK 11888, was designated as the neotype. A neotype acts as the replacement for a lost holotype. It becomes the new physical baseline for the species. SRC02, Ibrahim et al. 2014. But presenting a neotype from the Chemchem beds immediately triggers intense scientific scrutiny. Because of the extreme taphonomic sorting of the river system, and the lack of precise geological context from the commercial trade, skepticism is the necessary default. How do we know this isn't a chimera? A chimera, in paleontology, is a nightmare scenario where bones from multiple different animals, perhaps even different species, are mistakenly combined to create a creature that never existed. This skepticism is fueled by an active, ongoing debate over the taxonomy of the Chemchem Spinosaurids. Some researchers look at the subtle variations in isolated vertebrae and jaw fragments and argue that the region hosted multiple distinct species of Spinosaurids living alongside each other. They point to bones assigned to a genus called Sigilmassasaurus, suggesting that the Chemchem was a crowded, highly diverse ecosystem. Other researchers argue that these variations simply represent differences in age, sex, or individual variation within a single species. Spinosaurus aegyptiacus. When the taxonomy itself is contested, presenting a new skeletal reconstruction requires absolute rigor. To prove that the scattered bones of Fsak 11888 belonged to a single individual, the researchers could not rely on field associations. They had to turn to the laboratory. They placed the disparate bones, pieces of the skull, the spine, the pelvis, and the hind limbs, into an industrial CT scanner. CT technology allows paleontologists to look inside the fossilized bone without destroying it. What they are looking for are the internal markers of biological age. Like trees laying down annual rings, dinosaur bones exhibit lines of arrested growth. By analyzing the internal microstructure and the degree to which different bones have fused during the animal's lifetime, researchers can establish a precise developmental age. The CT scans of the neotype material told a consistent story. The internal growth markers matched across the disparate fragments. The bones belonged to a single, subadult individual, with the chimera fear reasonably addressed. The team could begin the process of scaling and assembling the digital skeletal diagram. When the 2D model was finalized, the resulting silhouette was shocking. We are accustomed to the standard body plan of giant theropods. Look at Carcharodontosaurus, or a tyrannosaur. They are balanced like massive, predatory seesaws. A heavy tail counterbalances a massive head, and the entire structure is supported by long, powerful hind limbs built for terrestrial locomotion. But the neotype of Spinosaurus broke that mold. The pelvis was surprisingly small. The hind limbs were drastically reduced in length. SRC02, Ibrahim et al. 2014. When these shortened legs were plugged into the digital model, the center of mass shifted dramatically forward. If you pictured this animal trying to walk on two legs across dry land, the mechanics become incredibly awkward. A terrestrial bipedal stance, while perhaps not impossible, would have been clumsy and energetically inefficient. The anatomy was pulling the animal away from the center of the continent, and dragging it toward the water's edge. This shift was reinforced by the anatomy of the skull. The elongated, crocodilian-like snout is riddled with a network of tiny holes, called foramina. In modern crocodilians, these pits house highly sensitive pressure receptors, allowing the animal to detect the tiny pressure waves created by swimming fish in murky water. The presence of this network strongly suggests that Spinosaurus was dipping its snout into the Chemchem rivers, hunting by touch as much as by sight. But skeletal anatomy, no matter how suggestive, has limits. A long snout with pressure sensors indicates an animal equipped to hunt in the water. Shortened hind limbs indicate an animal less suited for extended overland travel. These are compelling structural clues. But bone shape alone cannot definitively prove what an animal actually consumed. Anatomy suggests a lifestyle, it does not confirm the diet. To prove that Spinosaurus aegyptiacus was truly tied to the aquatic food web, we cannot rely on the shapes of the bones. We must bypass the structural mechanics entirely and look for the chemical fingerprint of the river itself. The chemistry of a river leaves a permanent mark on the animals that drink from it and feed within it. While the violent currents of the Chemchem delta shattered ribs and scattered vertebrae, they could not destroy the atomic signatures locked inside the teeth. Enamel is the hardest biological substance in the vertebrate body. It survives river transport far better than bone. And as an animal grows its teeth, It incorporates elements from its environment directly into the enamel's crystal structure, to bypass the debate over bone shapes and biomechanics. Researchers turned to a tool that deals strictly in absolute values, the isotope mass spectrometer. Isotopes are simply variations of an element. Calcium, for example, exists in slightly different atomic weights. The ratios of these different calcium isotopes vary between environments. The water in a flowing river holds a different ratio than the moisture trapped in the leaves of a terrestrial fern. When a fish absorbs calcium from the river, it locks that aquatic isotopic ratio into its body. When a dinosaur eats that fish, it inherits that aquatic ratio. Conversely, when a terrestrial herbivore eats land-based plants, and a predator eats that herbivore, a distinct terrestrial ratio is passed up the food chain, by grinding a tiny sample of enamel into dust, vaporizing it, and firing it through a mass spectrometer. Scientists can read the isotopic ratio. It provides a direct chemical readout of an animal's diet and habitat. But reading the signature of a single Spinosaurus tooth is useless without a baseline. You have to map the entire ecosystem. In a 2018 study, researchers extracted enamel from a wide cast of ChemChem survivors. They tested the teeth of giant crocodilians and the barbed rostral spikes of Onchoprestes, the massive prehistoric sawfish that choked the ancient waterways. They tested the teeth of flying pterosaurs. Crucially, they also tested the blade-like teeth of Carcharodontosaurus, the apex terrestrial predator of the region. SRC03, Hassler et al. 2018. When the data is plotted on a scatter graph, the results draw a sharp boundary across the ancient Sahara. The calcium isotope signatures of Carcharodontosaurus cluster neatly in the terrestrial zone. It was a land-based hunter, feeding on the few herbivores that existed on the floodplain. The crocodilians and sawfish form a tight cluster in the aquatic zone. When the enamel of Spinosaurus is plotted onto this graph, it does not sit in the middle. It does not overlap with the terrestrial theropods. The calcium isotope composition of Spinosaurus falls directly within the range of the aquatic taxa. It overlaps heavily with the giant fish and crocodilians. SRC03, Hassler et al. 2018. This is a profound reframe. The chemistry provides absolute proof, independent of any assumptions about shortened legs or pressure sensors, that Spinosaurus relied heavily on the river system for its food. It was not a rival to Carcharodontosaurus on land. It was harvesting the immense aquatic biomass of the delta. The presence of Onchoprestes barbs alongside Spinosaurus fossils is not just a coincidence of taphonomy. The mass spectrometer tells us that these giant, heavily armored sawfish were likely a primary prey source, but this chemical certainty introduces a new mechanical constraint. If your diet consists almost entirely of giant, fast-moving aquatic prey, you cannot simply stand on the dry bank and hope they swim past. You must enter the water physically. You must overcome the resistance of the fluid environment. This is where the anatomy of a standard theropod dinosaur fails. Dinosaurs are fundamentally built for land. They are buoyant. Their massive lungs and the complex system of air sacs that extend throughout their bodies make them float like corks. A standard theropod trying to hunt submerged fish would struggle constantly against its own buoyancy, expending massive amounts of energy just to stay below the surface. If Spinosaurus was harvesting the depths of the river channels, the chemistry demands that its body must have evolved a physical solution to the problem of buoyancy. It needed a way to sink. It needed a way to move. The evidence for these adaptations would not be found in the chemistry of the teeth, but hidden deep inside the architecture of the bones themselves. In 2020, fieldwork at the Neotype site in Morocco yielded a discovery that fundamentally altered the silhouette of Spinosaurus. Deep in the steep sandstone, the extraction crew uncovered a series of articulated caudal vertebrae, the bones of the tail. SRC04, Ibrahim et al. 2020. In most giant theropods, the tail acts primarily as a rigid counterbalance. held stiffly behind the animal to offset the weight of the torso and skull. The bones are robust, wrapped in thick musculature, but relatively compact in their vertical profile. The bones pulled from the neotype quarry were completely different. Extending upward from each tail vertebra was an extremely tall, strut-like neural spine, mirroring the massive sail on the animal's back. Furthermore, long chevron bones extended downward from the underside of the vertebrae. When these pieces were added to the digital skeletal diagram, the tail ceased to be a simple, tapering counterbalance. It formed a deep, broad, fin-like structure. SRC04, Ibrahim et al. 2020. The anatomy suggests a flexible, paddle-like organ, capable of generating lateral thrust in a fluid environment. It is a stunning visual confirmation of an aquatic lifestyle, but a paddle is only useful if the animal can actually submerge. And here, the inherent biology of dinosaurs presents a massive obstacle. As a group, theropod dinosaurs are incredibly lightweight for their size. If you look at the cross-section of a femur from a Carcharodontosaurus or a Tyrannosaur, you will see a thin outer wall of dense cortical bones surrounding a massive hollow center, known as the medullary cavity. This hollow architecture, combined with a network of internal air sacs, allowed terrestrial dinosaurs to grow to enormous sizes without becoming too heavy to walk. But hollow bones are a disaster in the water. They act like internal flotation devices. If Spinosaurus had a hollow skeleton, its massive body and air-filled lungs would have kept it bobbing helplessly at the surface, rendering its newly discovered paddle tail nearly useless for submerged pursuit. To prove that it was built for the water, researchers had to prove that it broke the fundamental architectural rule of its own lineage. They had to look inside the bone. In the lab, this requires destructive testing. A researcher takes a section of a Spinosaurus femur, mounts it under a diamond-tipped saw, and carefully slices through the 95-million-year-old fossil. The slice is then ground down until it is thin enough for light to pass through it, creating a histological thin section that can be examined under a microscope. When you place a standard theropod thin section under the lens, you see the dark ring of bone surrounding the large, empty white space of the medullary cavity. When researchers from a 2022 study placed the Spinosaurus thin section under the microscope, the empty space was gone. The bone is almost entirely solid. The dense cortical bone grows thick and heavy, filling in the center until only a tiny cavity remains. This condition is called osteosclerosis. SRC05, Fabry et al. 2022. This is not a pathology or a disease, it is a highly specialized evolutionary adaptation. We see extreme osteosclerosis today in animals like penguins, manatees, and early whales. It is ballast. By replacing hollow airspace with heavy, solid bone, the animal drastically increases its overall density, allowing it to counteract the buoyancy of its lungs. When the digital skeletal diagram is updated, color-coding the bones to represent this extreme density, the image of the animal shifts again. The chemistry proves it ate aquatic prey. The paddle tail provides the mechanism for thrust, and the histology, the solid, ballasted bone, proves it was built to sink. SRC05, Fabry et al. 2022. This cascade of evidence seems to paint a clear, undeniable picture. Spinosaurus aegyptiacus was an agile, submerged river monster, cruising through the depths of the Kemkem Delta like a reptilian submarine, hunting down giant sawfish with sweeps of its paddle tail. But paleontology is not just the study of bone shapes and bone density. It must also contend with the absolute laws of physics. And when the ballasted, paddle-tailed skeleton is handed over to biomechanical engineers, the water pushes back. To test how an extinct animal moved through water, researchers cannot rely solely on the shape of the fossilized skeleton. They must rebuild the animal in 3 dimensions, accounting for the flesh, the organs, and most importantly, the air. In a 2022 study, A team led by Paul Sereno constructed a 3D digital wireframe mesh of Spinosaurus, wrapping the neotype skeleton in a modeled volume of soft tissue. They factored in the extreme bone density revealed by the histology, adding the required mass to the limbs and ribs. But then, they had to program the lungs and the internal air sacs. SRC06, Sereno et al. 2022. This is where digital modeling encounters a hard constraint. Soft tissue does not fossilize in the Kemkem sandstone. We do not know the exact volume of a Spinosaurid's lungs, nor do we know exactly how their internal air sacs were distributed throughout the torso. The software must rely on estimates based on modern analogues, like crocodilians and birds, because air is vastly less dense than bone or muscle. The placement and volume of these virtual lungs dictate the center of buoyancy. If the center of buoyancy does not align correctly with the center of mass, the animal becomes physically unstable in a fluid environment. When the 3D Spinosaurus model is dropped into a digital water simulation, the results challenge the submarine hypothesis. According to the fluid dynamics calculated by the software, the high placement of the massive dorsal sail, combined with the estimated lung volume in the upper chest, creates a top-heavy structure. SRC06, Sereno et al. 2022. In the simulation, the model struggles to maintain a stable, upright posture in deep water. It is prone to tipping. It tends to roll onto its side. To prevent capsizing, the animal would have to constantly expend energy, using its limbs to right itself. This physics pushback creates an intense, active debate within the scientific community. If an animal is physically unstable in the water, it cannot be an agile, highly maneuverable pursuit predator. A predator cannot efficiently chase down a fast-moving sawfish if it is struggling to keep itself from rolling over. The fluid dynamics model suggests an alternative interpretation. Instead of a deep-water pursuit swimmer, the physics imply a heavy wader. In this scenario, Spinosaurus used its dense, ballasted bones not to sink gracefully into the depths, but to plant its feet firmly on the river bottom. By standing stable against the rushing currents of the delta channels, it could act as a massive ambush predator, dipping its sensor-pitted snout into the water to snatch fish as they swam past, much like a giant, heavily armored heron. SRC06, Sereno et al. 2022. This is the frontier of our knowledge. We have two sets of data locked in tension. The anatomical paddle tail and the dense bone histology suggest an animal highly adapted for submerged swimming. The biomechanical physics software suggests an animal that would capsize if it tried to swim like a crocodilian. We must apply a constraint card here. The fluid dynamic models are rigorously mathematical, but they are entirely dependent on the assumed volume of the lungs and air sacs. If the actual Spinosaurus had slightly smaller lungs, or could compress its air sacs differently than the software assumes, the buoyancy calculations change entirely. The software cannot perfectly replicate the biological reality of an animal compensating with living muscle and instinct. The physics cannot definitively prove exactly how the animal swam. But what the model does successfully accomplish is breaking the assumption of cinematic perfection. It forces us to realize that evolution does not always produce perfectly streamlined machines. Sometimes, adaptations are awkward compromises. A massive sail, a paddle tail, and dense bones might have made Spinosaurus a formidable predator at the water's edge, even if it lacked the elegant, torpedo-like grace of a marine reptile. Look again at the map of the late Cretaceous Sahara. Pull back from the isolated sandstone cliffs of Morocco and look at the vast expanse of the Kemkem Delta as it existed 95 million years ago. It was a world dictated by the flow of water. In the dry interior of the continent, Carcharodontosaurus hunted the sparse populations of terrestrial herbivores, its blade-like teeth engineered to shear through flesh and bone. But as the land sloped downward toward the Tethys Sea, the ecosystem transitioned. The river channels widened, choked with mangrove-like vegetation and teeming with giant lungfish, coelacanths, and armor-plated sawfish. This was the domain of Spinosaurus aegyptiacus. When we assembled the final, reconstructed silhouette of this animal, constrained by a century of accumulated evidence, we are not looking at a standard dinosaur. We are looking at a creature that evolution stretched to the absolute limits of its terrestrial architecture. It carried a massive, tail-backed torso on drastically shortened hind limbs. Its long, crocodilian snout was studded with pressure sensors to hunt in murky water. The enamel of its conical teeth locked in the chemical isotope signature of a diet pulled directly from the river. Its slim bones were thick and solid, the hollow cavities filled in with heavy osteosclerosis to provide ballast. And its tail was deep and flat, forming a flexible paddle. We know all of this because the physical and chemical evidence demands it. The laboratory tools, the CT scanners, the mass spectrometers, the diamond saws, and the microscopes, have allowed us to resurrect the biology of an animal whose bones were scattered by ancient currents and whose holotype was destroyed by modern war. But we also have to respect the limits of deep time. The final piece of the puzzle, the exact mechanics of how it moved through the water, remains elusive. The fluid dynamic models show us that extreme density and a paddle tail do not automatically guarantee stability. The physics of water displacement and buoyancy rely on soft tissues and air sacs that simply do not fossilize. Whether it was an agile, submerged pursuit hunter diving after sawfish, or a heavy, ballasted wader standing like a titan in the rushing channels to ambush its prey, is a question the evidence cannot yet settle. The exact swimming stroke is lost, and perhaps that is exactly how science should work. We push the methods to their absolute limits, extracting isotopic data from microscopic scratches of enamel, until we hit the boundary of what the Earth is willing to preserve. The Chem rivers are gone. The water dried up millions of years ago, leaving behind the sheer, baking sandstone cliffs of the Sahara. The bones that wash out of those cliffs are fragmentary and broken. But by trusting the chemistry and the physics, we have managed to pull the shadow of Spinosaurus back from the river's edge. Even if the depths of the water remain a mystery,